welcome back to Davin Biology. So this is our third and final part of our so-called mini series of state trials. So this third and final part, however, I will divide into part A and part B, because part A I will basically do section A, structure, and part B I will do section B and C, assay. So as you can see at the screen below here are all the chapters that are inside this state paper. So I will cover most of it. Okay, because now why Melaka paper? Because this paper actually I feel that there's quite a lot of important topics for this year. So quite a lot of like chapters that I feel have very high chance to come out this year are mostly in this paper. So these questions are very good. So we just do it as a revision. So if you see that any topics here that you need more help on, that you can skip to that part and watch whichever topic you think that you are quite okay already, you don't want to watch, also can, or you want to watch the whole thing, lagi best. Okay, so now first question, Form 4 Chapter 2. So this Form 4 Chapter 2, besides organelles, what the amoeba, paramecium, unicellular organism, right, there's this thing called tissue organization, cell organization, which is about different cells and tissues in human. Okay, so the first thing that they quite like to ask, and still a lot of you don't know how to answer this. Now they always ask this, name the cell organization level. So what's cell organization? It's a very simple thing. Since you learn it since Form 1. Basically it's the cell, tissue, organ system, the organization. So X is an organ. This is your small intestine, right? So that's how you answer this. Third level, cell, tissue, organ. So quite often, let's say if they ask the amoeba paramecium question, right? Amoeba and the paramecium, if they ask you this, what is the level of cell organization of amoeba, of paramecium? So since they are unicellular organism, they are basically just a cell. Okay, they don't form tissue, they don't form organ. Right? A2, name the main tissue that forms the whole of structure. X. So for our internal organs, right? Actually, all of our internal organs, the surface always have this surface tissue, the skin punya tissue called the epithelial tissue. Easy. And next, okay, I already written the answer here. One adaptive feature found in structure X to absorb nutrients efficiently. So they want the adaptation of the epithelial tissue in our small intestine. So since it's to absorb nutrients, now the epithelial tissue of our small intestine are basically the villus punya wall. So if you remember, right, okay, let's say here's the villus. The wall of the villus, right, have a lot of these epithelial cells, which have a lot of tiny hairs on top of it, which are called the microvillus. And also, right, they are one cell thick because nutrients have to diffuse into it easily. So one of the key features is very thin. How thin? One cell thick only. Okay, so that's the first thing. So if it's one cell thick means very thin, it increases the rate of absorption of nutrients. Like I said just now, the nutrients can diffuse easily. And with all these tiny hairs, right, called the microvillus, right, it increases the surface area for absorption. What do you mean by increase the surface area? Okay, you see, if let's say this is our epithelial, so without the microvillus, the surface is just like this. But let's say with the microvillus, the surface now is like this, is bigger and wider, so nutrients can go into it faster lah. Okay, so minus microvillus, so increase surface area for absorption. So you have more area, your rate of absorption is definitely faster. Moving on. Now, cancer. Suddenly you talk about cancer, so this is more of like a Form 4 Chapter 6, mitosis, meiosis chapter. Cancer starts when tissue in structure X divides uncontrollably. Explain why cancer easily starts on tissue found on structure X. Okay, so since epithelial tissue is the surface tissue are more easily exposed to those kind of so-called carcinogenic substance, substance that cause cancer. So since they're on the surface, they are more exposed to this stuff. So let's say anything that you eat that is carcinogenic. For example, you eat a lot of artificial coloring, flavoring stuff, a lot of barbecue, those kind of black burnt stuff. So the surface of your small intestine are the first cells and tissues exposed to this substance. So that is the reason. So it is the outermost layer in the organ. So hence, you can also say it will be more easily exposed to what we mentioned just now as the carcinogenic substance. So these cells, they will mutate more easily and divide by uncontrolled mitosis. Okay, so you can say causing it to mutate more easily. Yeah, so this one, nothing too difficult. The first thing you have to say is the outermost layer more exposed lah. Moving on, next question. Okay, this question, I won't do this question because if you look at my part 1 state trial series video, actually I got do this one, Form 4 chapter 4, especially I did this part, the DNA nucleic acid one. Yeah, so this one, I won't do that, it's very simple. Okay, so this one you can try yourself. So I want to spend more time on tougher questions, like okay, question 3, Form 4 chapter 5, enzymes. So now this enzyme question is a bit different than what we usually see, okay, which I think this kind of question have a chance to come out, so better we know about it. Now, this is actually called, what we call as an enzyme immobilization. Almost forgot the name. Okay, so this is a technique used in industry, so I just write here, enzyme immobilization, which mobile means move, 可以动. Immobile means now the enzyme cannot move. So this, how they make the enzyme unable to move, right? Okay, they made it into these bits, this round thing called bits, 些珍珠. So each of these bits inside got this enzyme K, which is used to actually convert normal milk to lactose-free milk. So if your bio basic is quite well, you should know that this enzyme K should be lactase because it can break down the lactose in milk to become lactose-free. So lactose, by the way, the sugar in milk, 乳糖, broken down by this enzyme lactase to become glucose and galactose. So that's basically what's inside the lactose-free milk right now. So this enzyme K is lactase, break down the lactose. So, okay, I've done the first question for you already, lactase. Name the technology used in an industry. This is called enzyme immobilization technology. Moving on. Then after that, right, in enzyme immobilization question, right, for this one, they always like to ask the benefits, the advantage of this enzyme immobilization, especially this enzyme K, these bits, right? So how did this allogeneic bits help the activity of enzyme K? So what's so special about these enzymes? Number one, these enzymes, right, since they are in the form of like bits like that, right, when you pour the milk, right, the milk, of course, it's liquid, can go through the gaps and then come out from these bits easily. Meaning to say, the milk, the product, the lactose-free milk, can be easily separated from the enzyme. Takkan you want to drink with the enzyme together, ma? So the enzymes are made into this form so that it can be easily separated from the milk. That's the first thing. Okay, it can be easily separated. And the next thing I already mentioned here, enzyme reaction will remain in the same position throughout the process. Which is enzyme K, mistake here. Which means this enzyme, right, it won't move. If the enzyme keep moving around, right, you have to take it out again and then put it back in the position. So since it's in fixed position, right, I just have to keep on pouring the milk. Okay, done already, then take another one, pour. So it saves a lot of time because it won't move. And the third and final thing you can say, right, just like any other enzymes, enzyme does not break down after the reaction, which means this enzyme can be reused repeatedly. You don't have to keep on making new one. So that is one of the most common thing about enzyme. It can be reused repeatedly, so we all know already, right? Okay. C. Diagram 3.2 shows a type of plant used by a housewife to cook meat. So by the way, this did they mention here? Okay, they didn't mention, but it's obviously a papaya tree. So for papaya, there's only one use in this enzyme thingy. So if you see papaya, the enzyme here definitely involved here is papain. So let's read here. They say, wrap the meat slices in young leaves and marinate for several hours. Marinate is washed before cooking. Okay, so they use the papaya leaves. They didn't tell you this papaya, but yeah, it's papaya leaf. They marinate with meat, and then after that already, they remove the leaf and then cook the meat. So this papaya leaf, right, has an enzyme called papain. Okay, so you can say the leaves, which is the papaya leaves, okay, contains an enzyme called papain. Which papain is a kind of enzyme that can break down the protein structure in meat. So it's like sort of like a protease like that. So this is to tenderize the meat, make the meat softer because you break down the protein structure inside the meat. So that's what you have to say, which, okay, don't use the word breakdown. Always when we break down a complex structure to a simple structure, we will say hydrolysis, the protein structure meat. So the main thing you have to say is, this is to tenderize the meat, act as meat tenderizer. This is to tenderize the meat. That. For question number 4, this is a Form 5 Chapter 8 question, biodiversity. Now for this chapter, overall I think the challenges are not very high, but let's say if this chapter were to come out in our 2024 SPM, right, I think that the most likely topic they will ask is this nitrogen cycle, because this part haven't came out yet. Okay, for this cycle, I think I will go through a bit first. Now, nitrogen cycle basically is a process that occurs in soil, and plants rely on this cycle a lot because plants need nitrogen a lot. It's very important for plants. But plant cannot directly absorb nitrogen gas from the atmosphere. It only can absorb nitrogen in the form of certain ions, which these ions are only produced through the nitrogen cycle. Not only, but are mainly produced through the nitrogen cycle. So the first process, in red color here, is called nitrogen fixation, whereby nitrogen gas is converted into ammonium, which is NH4+. Let me write it down here. Okay, which is carried by a bacteria called nitrogen fixing bacteria. So this is what you need to know. There are processes here, 3 processes, I forgot to mention this earlier, which are carried by 3 different bacteria. So we just mentioned the first one. So example of nitrogen fixing bacteria are rhizobium, which in a while later the question we will mention this, what I actually done. So the next process is, right, the ammonium then will be subsequently converted into nitrites, NO2- and nitrates, NO3-. So this process is called nitrification, which is carried out by nitrifying bacteria. Now there are 2 examples of nitrifying bacteria, Nitrosomonas, which is the first one, and Nitrobacter, which is the back one. And the final one, the final process, right, is actually to return the nitrogen back to the atmosphere because it's a cycle. So this is called denitrification, where I break down the nitrate into nitrogen gas and oxygen gas. This is called denitrifying. This is carried out by denitrifying bacteria. However, there's no example for denitrifying bacteria. Now, if you go back to the first process, right, ammonium also comes from decomposition of dead organisms. So when animal dies, when plant dies, when it's being buried in the soil, the body of the animal, for example, will be decomposed by, let's say, bacteria, decomposes to form ammonium, which then contributes to this cycle. So decomposition actually is, contribute to this nitrogen cycle as well. However, right, in nitrogen cycle, right, the decomposition process to produce ammonium, right, is, we don't call it as decomposition here, we will call it as ammonification. So take note, if they ever ask what is the process of getting ammonia from dead organisms, in this cycle we will mention it as ammonification. Okay, so back to our question just now. So you can see the cycle begins from here, nitrogen gas. So then this nitrogen gas is act on by this nitrogen fixing bacteria to actually produce NH4+ ammonium. Okay, and then only this nitrifying bacteria convert the NH4+ into nitrites, NO2-. Now at the same time, you can see ammonium, NH4+, also comes from dead organism, which they then label here as process P. So we mentioned just now already, right? The process of decomposing dead organisms to get ammonium is called ammonification, not a decomposition for this nitrogen cycle. And then NO2 continue to become compound Q, which is of course NO3, which is then, they say here, absorbed by plants. So actually plants not just absorb NO3 nitrate, it also can absorb nitrite, NO2-, and also ammonium, as we mentioned earlier. Okay, and then you can see this NO3 returns back to nitrogen gas. So that's the cycle. Alright, so yeah, Q is basically nitrates. You have to spell the whole thing out. Yeah. Now, state an example of organism involved in process P. So basically, you just have to name a decomposer. Bacteria is the most common one. Funguses, most mushrooms, fungus, they are decomposers as well. And then the next question. Okay, predict what. Okay, before I do this question, I think we talk about this leguminous plant a bit first. So there's something about these legume plants, right? Okay, legume, right? They are basically bean species plants, like papain. Okay, beans, toh. So these plants, right, their roots, let me just draw here a bit, okay, their roots, as these are roots, right, have like pimples like that. called root noodles. Now, inside these root noodles, right, the pimple part of the roots, right, there's this bacteria that live inside it that we mentioned just now earlier, called the rhizobium. So rhizobium is an example of nitrogen-fixing bacteria which converts nitrogen gas into ammonium. So if they ever ask you what's the example of this bacteria, there you go, we mentioned just now already, rhizobium. Okay, then continue this question. They say, predict what's happened to the ecosystem if organisms stated in B1 is dead. So if there's no decomposers, all the dead body in this world cannot be decomposed, right? So decomposition does not occur. Now the main problem with this is that the nutrients in the animals or the dead organism cannot be returned back to the soil. Now why returning nutrients in dead body back to the soil is important, because the food chain, right? You have producer, plants, right? They are being eaten by primary consumer, then secondary consumer, and then tertiary consumer. So you can see this food chain, it's a bit unfair to the producer, because producer produce for everyone, but no one return them in favor. So basically when they die, decomposers will decompose their dead body into what we call as ammonium, just now remember. So when they become ammonium, NH4+, now that is how then plant can sort of like indirectly eat them more. So it's basically to balance the food chain, to make sure plants get enough nutrients. So as you see, nutrients in dead organisms are not returned to soil. So basically, you also then can say there will be less ammonium in soil for plants. Now, other than that, what will happen to the ecosystem also is that pollution. Dead bodies will power up and it will never be decomposed. So you can say the dead, you can say that it will cause pollution, okay, as dead organisms will not be decomposed. Okay, basically the food chain lah, you can say the ecosystem will be imbalanced as well. Okay, then this is about the leguminous plant, as we were mentioning earlier. So a farmer grows leguminous plants as rotational crop in his farm. Explain the importance of this practice. Now, what do you mean by rotational crop? So let's say we have a plot of land. Okay, so let's say they divide into 4 different plots. Okay, so let's say here I plant cabbage, 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 but there'll be one plot, right? They'll reserve it for me. Let's say they plant the leguminous plant. Okay, so let's say after this, all this cabbage, they harvest already. Then these leguminous plants, they'll shift over here. And then they plant again, cabbage here, cabbage here, cabbage here. So they will keep on like rotating the leguminous plant. And after that, these leguminous plant are plant here, then the cabbage all move here like that. Okay, so why do you want to do that is that it's because we mentioned leguminous plant, there's the root noodles inside called rhizobium. I mean, this rhizobium can convert nitrogen gas into ammonium. So this will help to increase the ammonium content in the soil, make it more fertile, more nutritious for plants, for crops that you want to grow later. Okay, so this is what we can say. Talk about the root noodles, talk about the rhizobium. So you can say because its root noodles contains this bacteria called rhizobium sp. Okay, so you have to remember the name of this bacteria for this question. Okay, so you can say which convert nitrogen gas into ammonium. If you write the process, also you get 1 mark through nitrogen fixation. So the purpose is to increasing fertility of soil. Oops. Alright, so that is the very important thing about leguminous plants. So sometimes you will say, as a farmer, what can we do to increase fertility of soil? Now, planting leguminous plant nearby your crops is one of the easiest things you can do because of the root noodles. You've got rhizobium, convert nitrogen to ammonium. Okay, moving on. Form 5 chapter 2. Now, one of a very, very important plant chapter for this year SPM, because this is one of the plant chapter, or should I say the only plant chapter that have not come out yet in our KSSM SPM. So this one, you have to know that. Okay, so leaf photosynthesis, for example, quite important. So for this question, they ask us to complete 5.1 by drawing the layer of palisade mesophyll cell. So palisade mesophyll cell will be always right below the upper epidermis, and they are vertically arranged punya cell. I don't know how to draw this, I covered up already, so I just draw half of it, like that. Okay, then of course you can draw the nucleus, okay, the chloroplast inside, the vacuole. So nucleus, chloroplast, agak-agak. This one, the main thing, you just have to agak-agak only because they want you to know where's the position, then can already. Okay. Now explain the adaptation of palisade mesophyll cells for photosynthesis. Now, so this is how I will teach my students to remember palisade mesophyll. Okay. So remember these cells, they are very sad. Why sad? Because they are like people that have to squeeze into LRT during rush hour. Okay, during 6 p.m. rush hour. So in LRT 6 p.m. right? Okay, train or buses or public transport, any public transport, be very packed. So when they pack, you squeeze with, imagine you have to squeeze with so many people, this is very sad. And when you squeeze with people that are in this public transport, there's no place to sit, so everybody have to stand vertically. So you just remember, palisade, very sad because it has to stand, so they are arranged, we will say vertically, and closely packed. So when they pack and stand up, that's why they're very sad. That's why the cells, they're all vertical. See, they are not like horizontal, they're all standing up one. So they are packed, arranged vertically and closely packed. Now, this is for basically for them to absorb maximum sunlight. 2 marks, easy done. So how does this actually help them to absorb maximum sunlight? Now, we don't need to explain additional things. This is good enough. But if you want to know, very simple, because if let's say all the palisade mesophyll, there are gaps between them, they're not packed closely. Sunlight, will go past them like that. So there will be light that they would not absorb. So they will not absorb maximum sunlight. And if let's say they are lying down, they will take up more space, meaning you can fit lesser cells, resulting in lesser sunlight can be absorbed. Just like in LRT, in the bus, if everybody lie down, you take up more space, you can fit in lesser people. Same thing. Now, next, C. A farmer finds that his tomato plants produce fewer fruits when grown in shaded area. Of course, if there's no sun, how to photosynthesis, right? How is he even a farmer? So dumb. So you will say, of course, there's low light intensity, leading to low rate of photosynthesis. So why low photosynthesis? The fruits, the tomatoes will be smaller because photosynthesis produces sugar, glucose. Glucose, the sugar forms the fruit. So basically lesser glucose produced for development of tomatoes. Tomatoes are fruits basically. Okay, so fruits needs sugar. Moving on, compensation point. What the question here asks, based on the graph, explain what happens to the rate of glucose production when light intensity continues to increase beyond compensation point. Okay, this is so easy. So, light increase, photosynthesis increase, because you can see the intake of CO2 increase. So, more light, more photosynthesis, that's why they take in more CO2 because photosynthesis uses CO2. So, as the light increases, rate happens to rate, okay, they want rates of glucose production. So, let's answer this first. Of course, the rates of glucose production increase. More photosynthesis produce more glucose. Why? So just explain because rate of photosynthesis increases. So for this one, the explanation, you don't have to talk about carbon dioxide like what the graph shows, because read the question, rate of glucose production. Okay. Now, and just to add a bit more things here, so they didn't ask the main thing about compensation point. So usually, if they ask, explain what happens at compensation point. So basically over here, your rate of photosynthesis is the same as rate of respiration. Meaning to say, the rate of CO2 intake is also the same as CO2 release. So you can just follow this. Okay, intake. Okay, I just write here, CO2 intake is the same as CO2 release. Glucose production, glucose consumption, also the same, because these two processes is the same. Form 4 chapter 12, a very important chapter as well, which I think this chapter also have chance to come out in essay, but however, today we're not doing essay for this Form 4 Chapter 12. So this is a neuron. Name the part label X. Okay, before we do this, I think we state the type of neuron first. I think that's the most important thing. You must determine what neuron is this. So we have three types of neurons, right? Sensory, relay, motor. So this is actually very obvious, a sensory neuron. If you're asking why, it's because the cell body is located in the middle of the neuron. For motor neuron, the cell body is at the end part, we call it the terminal, and it will look something like this. Okay, so that's the difference. Part label X. So in a neuron, right, the structure is like human. There's head, there's body, there's tail. So this is the head, it's called the dendrite. So X is the dendrite. Okay, so body is called the axon. So, okay, for dendrite, right, it will always connect until the cell body. So even though here is look like the head only, but actually the head is stretched all the way until the cell body. Then the subsequent part here, or only we call it is the body, which is the axon, which is just this axon. And the butt, where the signal will then come out, is called the synaptic. However, for relay neuron, for motor neuron, right, dendrite is only here. Because the cell body is located right at the head over here. So therefore, this whole thing will be the axon. Then synaptic knot will be behind here. This one done already, sensory neuron. Draw an arrow in the box to show the direction of impulse. Oh, the box is supposed to draw the arrow, okay. So the box is supposed to draw the arrow. Of course, you move from head to tail lah, this direction. Okay, how do I know where's the head, where's the tail? Because synaptic knot, okay, if you know the meaning of knot, knot means like door knot. You know the door that you use to twist that part. So there's this round thing you can see here. So that's the synaptic knot. See here don't have the round thing one. That's why this is not the synaptic knot, this is the head, the dendrite. So that's how I know. That's why the direction will be from left to right lah. Moving on, a patient has abnormality of the immune system that attacks structure Y in the brain. Explain the effect of the abnormality on the activity of impulse transmission. Now, Y, by the way, it's called the myelin sheath. Now, this thing is like the insulator of the axon. It helps to protect the axon. Now, this thing is like the coating of the wire like that. It doesn't just protect the wire, but it insulates it. It doesn't let electrical impulse to come out to shock your body. And on top of that, if the impulse doesn't come out, the impulse can flow transmitted faster. Okay. By the way, this disease is actually called multiple sclerosis. But this question, they didn't ask you to name the disease, right? They just asked you to state the effect of the abnormality. Right. Number one, without this, right, nothing to protect the axon. So the axon will be not protected, meaning to say it will get infected easily. And number two, now your nerve impulse, can come out. So since they say this occurs in the brain, transmission of impulse to the brain will be slower. And also your brain cannot transmit the nerve impulse out. So your movement, your reaction, all will be more slower and delayed lah. So that's what we can say. Okay, you can say, and then you can say also say less nerve impulse, such as short form NI, nerve impulse, transmitted to effectors. So you can say his reaction will be slower. You can say this also. He will have slower reaction. Okay, next one. This is actually hormone imbalance. So hormone is also related to this chapter. So individual A, normal individual, and individual B. So compare the individual in diagram 6.2 based on the following criteria. So hormone imbalance. So for A, he's too big. So basically he has excess growth hormone. This fellow will have lack of growth hormone. So effect on growth, if you lack of growth hormone, you will have stunted growth. If you have excess, you will have abnormal growth, means your growth is too much. Okay, or you can say excess, grow excessively also. Okay, so very simple. Okay, this is a growth hormone problem. And by the way, the condition, okay, let's just name the condition as well. So if, yeah, those kind of dwarf, very, very small adults, let's say they are adult already, but let's say they are not even 1 meter tall. So this is actually called dwarfism. And too much growth hormone, you'll be over 2 meters, over 2.5 meters, right? Those kind of very, very tall, giant people. Yeah, so it's called gigantism, which comes from the words giant. Moving on. Okay, last 2 structure questions, then we'll end our part 1 already. So for diagram 7.1, this shows the mechanism that occurs in blood when a person is injured. So this is actually Form 4, Chapter 10, the blood clotting part specifically. So now here you have something called activator R, which is something that works together with calcium ion and vitamin K to convert prothrombin to thrombin. So this R, it's the activator that sort of activates our blood clotting process. Now this thing is actually called thrombokinase. Now thrombokinase is actually released by platelets. You know, every time we say platelets are involved in blood clotting, now let's say you have a cut, let's say this is your blood vessel, botched already. So when platelets, they stick here, right, they will release this thing called activators. These activators called thrombokinase. Which thrombokinase will initiate this whole process to completely clot your wound. Okay, so it will convert prothrombin to thrombin, which we will say prothrombin is an inactive plasma protein to become active, because you want to clot blood, now you have to be active. So you can move from pro, become no more pro already. So when this thing is active already, it will convert another thing called fibrinogen to fibrin, which is a soluble plasma protein, to an insoluble plasma protein. So this insoluble thing does not dissolve in water, does not dissolve in blood. It's like we call it a thread-like protein structure. It's like a string like that, which will form like a net over your wound to seal the wound. So this is what will be formed at the end of the blood clotting process. Okay, so then the question that they asked here, fibrin, as I mentioned just now, is a thread-like protein fiber that forms a network, exactly like I mentioned, on the wound, on the surface to trap erythrocytes. Erythrocytes is your red blood cell. Explain the importance of fibrin in the mechanism of blood clotting. So the importance of fibrin is, basically this question, they are asking the importance of blood clotting, because for fibrin, what does it do? They already mentioned here. It's a thread-like protein fiber, forms a network, trap the erythrocyte. Like literally, the main function of fibrin they already mentioned, so you cannot repeat this already. So therefore, the importance of it is just basically the importance of blood clotting. So this is to prevent excessive blood loss. And besides that, you also want to prevent, you want to maintain your blood pressure, you want to prevent microorganisms from entering. You also can say that, basic importance of blood clotting. So I just read 2 very simple ones to maintain blood pressure. Okay. Next, this is disease, which, they say here, shows a condition. So there's a blood clot and a plaque. So plaque is formed from deposition of cholesterol. Blood clot, basically there's a disease whereby your blood can clot within the blood vessel. So this is actually called thrombosis. So now combo, 2 of these things happen together. So this blood vessel should be very, very blocked already. So they say this blockage occurs in artery S, which is called coronavirus, the coronary artery. Now this blood vessel is the main blood vessel that supplies oxygenated blood to our heart muscles, to our cardiac muscles. So if it's blocked, now that is one of the reasons that causes heart attack in people. So why people that have high cholesterol have heart attack, because this blood vessel could be blocked by plaque, by cholesterol, and it prevents oxygen from reaching your cardiac muscle. So the effect here, heart attack. We can start with that. Okay, now if you know the scientific name of heart attack, myocardial infarction, go ahead. To sound more cooler, it's accepted also. But yeah, you can just stick to heart attack. Then what will happen now? The oxygen cannot flow. So you can say because this prevents oxygenated blood from reaching your cardiac muscle. From being transported to your cardiac muscle. So your cardiac muscle, no oxygen. Muscle needs oxygen so that there's energy to contract, to pump. This cannot occur. So basically, your cardiac muscle cannot contract. So therefore, heart cannot pump blood to body cells. So that is the effect. You get lack of oxygen, and that's how people can die lah. Right, the next one. Explain the function of device T in overcoming the problem. So what is this T? Okay, so this device T is the device to replace the SA node. So some people, their SA node cannot generate electrical impulse to stimulate the contraction of their cardiac muscle. So basically, yeah, their heart will suddenly stop. Sometimes it's due to failure of this SA node. Not necessarily, it's like just now this blockage of your coronary artery. So this device T, we call it an artificial pacemaker. Works the same as SA node. So basically the question here is like asking the function of SA node, because how does this thing works? It works exactly like SA node. So first you can say, oh, this device T acts as an artificial pacemaker. And what does it do? Just like the SA node, it generates electrical impulse, okay, to stimulate cardiac muscles to contract and relax. Okay, you can add this word called rhythmically. So like how a normal SA node works like it stimulates your heart to pump in a certain rhythm, doop, 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 like that. Oh, that's it. Mention what is it? An artificial pacemaker, generate electric impulse, just like the SA node, stimulate your contraction of your cardiac muscles. Okay, last and final question for our structure. So this we end our part 1 with this question. This is a Form 5 Chapter 11 question, inheritance, which inheritance this year should be very high chance as well, because inheritance have not come out in SPM yet so far. So this question is a dihybrid cross, means you have two characteristics, two pairs of alleles. So for this question, they cross these two plants, inflated and round seed, cross with constricted pod and constricted seed. So they really give you the alleles here already, which we go into the question lah. So both of these plants, they are pure breeds. Pure breed means the allele got no mix. Is capital or small. Okay, no big small. So inflated pods and round seeds. So inflated is a dominant allele, round is also dominant. So since it's pure breed, it's capital F, and big R. So this is the opposite, constrict pod, constrict seed. So recessive definitely must be pure breeds, because the only way to show recessive is by having two recessive allele present without the dominant. So the gamete produce one F, one big R, one small f, one small r. Now, why the gamete is like that? Okay, because now why the gamete won't have FF and RR? Because in a pair of chromosome, okay, big F, let's say here's big F, okay, and then here you have R. So inside the cell, when they want to form gamete, then they will split like this. Meiosis that time. So F and F can never end up in the same gamete. So F must always pair with another allele from another pair. Therefore, you only can form with R. So since both are a pure breed, there's only one possible combination here, which is capital F, capital R. Same with the recessive one. So this is the offspring, the genotype. You will get, or we call this a hybrid, heterozygous. Big F, big R, small r. So we done this already. State the phenotype of F1 generation. So remember, phenotype means the physical characteristics. Genotype means the gene, the allele in the gene. So you write letters when they ask about genotype. Phenotype means what is this characteristic. So since both have dominant, F and R, F dominant is inflated, R is dominant for round. So therefore, this is inflated pod. Right, it's pod, right? And round seed. Next, I already draw the answer for you here, because Punnett square very easy lah. So construct a Punnett square of self-cross between offspring of F1 generation to form F2 generation. Okay, now so for plants, right? Okay, for this not plants, dihybrid inheritance, which we only do for plants, right? This is what they usually will ask. So in the first generation, right, they will cross a homozygote dominant and a homozygote recessive, or purebred dominant, purebred recessive, the same meaning. So in your F1 generation, you will get heterozygote, big, small, big, small. And what they will ask you later is, right, now if I cross all the off-1 offspring together, so plant, they can reproduce among themselves, right? Siblings can reproduce with siblings for plants. So they will ask you, if I cross this, what will be the F2B? Okay, so this is the answer. So now I cross this big F, small f, big R, small r cross with the same. So when big small cross with big small in dihybrid means the heterozygous cross with heterozygous, right? Okay, this is how we get the gamete. So you just combine all the possible F and R's. So basically, big F can combine with big R to form a combination. Big F can combine with small r to get this combination. Then small f can combine with big R and also small r. So it's like 1, 3, 1, 4, 2, 3, 2, 4. So that's the combination of the gametes. So for this Punnett square, you just have to write the gamete for the first parent and the gamete for the second parent, and basically when they cross like that, you get all of these. Okay, this one I won't go through right? So it's quite easy to do yourself. The next one is explain the ratio of the offspring produced. Now, so for dihybrid cross, I just memorized the ratio. It's 9, 3, 3, 1. Meaning to say you will get 9 of dominant characteristics, round and inflated. So for the 2, 3 is combination of dominant recessive. So when you mix with recessive, you will inherit lesser already. So big F small r and small f big R is 3 each, and for recessive only one is the least. So that is the first thing you have to say. The ratio you will get is 9, 3, 3, 1. That's it. You don't have to write anything else already. Then what is the Mendel's law involved? So for dihybrid is involving Mendel's second law, whereby Mendel's second law, you just have to explain during formation of gametes. Okay, now they say here, each allele from a pair of allele combine randomly with any allele from another pair of allele. Meaning to say, why when I cross these two, we call as heterozygous, right? Why would I get so many combinations? Just now when I combine dominant recessive, right, I only get one combination of offspring. Why now I get so many different combinations? Because during meiosis that time, okay, formation of gamete means during meiosis that time, okay, this is what will happen. Okay, this is called the independent assortment. So during metaphase 1 that time, when they align at the center of the cell, at the equatorial plane that time, now they have no fixed position. Meaning they can align at this F and F side by side, big F, small f, big R, small like that, or they can terbalik at that, see. So the position is not fixed one. The R you can see can be big R, small r, or the other way around. So if the position change, right, the combination of gamete also will be different, because if I align like this, right, the first combination, I will get big F, big R, small f, small r. But if I align like this, I will get big F, small r, small f, big R. You can see the combination different already. So this is what the Mendel's second law here is stating, which is each allele from a pair. So let's say this big F can combine randomly with big R, or even small r, which is from another pair of allele. So each pair from an allele, let's say that each allele from a pair, so let's say this big F, it can combine randomly with this or this, which is from another pair. So that's what Mendel's second law is taking. That's why we can get 4 different gametes, and that's why we get so many combinations in F2 generation. Okay, so this one you just have to write this, that's it. Now, this one will seem a bit difficult, but then you don't have to explain a lot, it's just like that only. Okay, so I will end our part 1 video here. So part 2 will be essay lah. Okay, so see you guys very soon. Bye bye.